Okay, so coming back to seed plants. So a couple things here. So seed plants, number one, have seeds, obviously. Seed plants are also vascular plants, other thing to know. And seed plants are all what we call heterosporous, which means we make two kinds of spores. Large megaspores, which will produce megagametophytes, and smaller microspores, which will produce microgametophytes. In the non-vascular plants, the plants are what we call homosporous, meaning we make one kind of spore that produces one kind of gametophyte. In the seedless vascular plants, we had some members that were homosporous and some members that are heterosporous. The members we looked at in lab were homosporous, so don't worry about those heterosporous ones for purposes of the lab practical. But for the seed plants, you do need to know that everybody is heterosporous. Megaspores, microspores. And we're going to have different structures associated with those things. So we actually only looked for the gymnosperms, which means naked seed, because they are not enclosed by fruits. There are multiple phyla. We only really looked at one phyla for this group. And so we're going to talk a little bit about those structures. So first of all, you probably know that conifers have cones. That's where they get their name from. But we have separate male and female cones. The female cones are sometimes, they get a few different names. Sometimes we call them seed cones. Sometimes we call them ovulate cones. We also call them a megastrobilus. Right? We've seen that word strobili before, right? A cluster of sporangia. In this case, it's called a megastrobilus because we have separate megastrobili that will produce megaspores and microstrobili that will produce microspores. And in the male, sometimes called microstrobulus. Let's look at them in cross section or long section so we can actually see internally what is going on. So let's start with the female structure, the megastrobulus. Okay, so this is really not different than the other strobili we've looked at, except the seed plants in general. Here, the gametophyte generation is so reduced, it's pretty much going to be microscopic. Now, it's still going to be multicellular, but it's very, very tiny. And you wouldn't even, notice it unless you basically would have to do some dissection pretty much. Okay, so if we look at the pine, and that's just the genus that is the example, the Pinus megastrobilus, we have a little leaf-like structure that we're calling the sporophyll. And then we have the ovule here. Okay, so within this ovule, we will find the megagametophyte that will produce the eggs. If we look over here at our microstrobulus, or our what we might call a pollen cone, each of these sacs here is a microsporangium. And by the way, this is our megasporangium. Within these microsporangia, we have the male gametophyte, aka pollen grains, that will be producing sperm. Let's take a look at some of this pine pollen. Okay, so conifers are wind pollinated. And so one of the things that we see, at least in this genus, is that we have these air sacs on the pollen that help with wind dispersal. Another important thing to understand about seed plants is that we now have a really important structure called the pollen tube. So this, if we were to look at the inside of this pollen grain, we would see it's not just one cell, it's multicellular. And one of these cells is called a tube cell. And this tube cell is going to generate a pollen tube. And what is so awesome about this pollen tube is it is going to be now the mechanism of delivery of the sperm to the egg. So we no longer need water. We don't have flagellated sperm anymore. They're going to swim through water. Now we have this way to get sperm to egg, and we don't have to rely on water. And in addition to having an increasing dominance of the sporophyte, another trend we see in the evolution of plants is an increasing independence from water. And the seed plants, one of the things that made them so successful is that their pollen, the structure of their pollen and these pollen tubes allows them to be independent from water for their reproduction. They don't have to have a droplet of water. That's a really big deal if you want to be terrestrial, right? Because there's not always water readily available. Now, yes, they're a plant. They still need water to live, of course, but they don't need it to complete their reproductive cycle, and that's a pretty darn big deal. Also, within the pollen grain, we're going to find cells that are going to give rise to the sperm. So sperm cells will also be produced, and these will be traveling down this pollen tube to reach the egg. Okay, so let's talk about phylum Anthophyta. So phylum Anthophyta, those are our flowering plants. They also have pollen, okay? And these pollen make pollen tubes that they'll be sending to sperm down. So that's all similar to, and in fact, a lot of things about their life cycle is similar to the conifers, right? They're heterosporous. They're going to be producing megaspores and microspores. They're microspores, aka the pollen grains, but their megaspores, I'm going to jump ahead for a second, are going to be found, and I can't find the structure I want, are going to be found in female structures known as ovaries. Well, actually, they're going to be found if we cross-section through this ovary. So here's a cross-section through an ovary. We find these structures called ovules, aka our megasporangium, where our megagametophyte will be producing an egg. Okay, so I just wanted to bounce back and say that is different than the conifers. Also, the name angiosperm roughly translates to housed seed or vesseled seed, and is because our ovules are enclosed within an ovary wall, and this ovary wall will eventually ripen to become a fruit, whereas the ovule eventually ripens to become a seed. Okay, angiosperm pollen, similar deal. All right, we're going to germinate a pollen tube that's going to contain two sperms. So we have a tube cell within our pollen grain that makes the pollen tube, and then we have two sperm. One of these sperm is going to fertilize, is going to join with the egg cell to form the zygote, and one of these sperm is going to fuse with the polar nuclei that we find in what we call the central cell. And this is going to give us the endosperm. And this is another big important difference between gymnosperms and angiosperms. This is called double fertilization. And it's kind of a big deal. The endosperm is going to be the food source for the developing embryo. Okay. Another obvious difference between angiosperms and gymnosperms is that angiosperms have flowers, which is why they're pretty, and we give them to each other for Valentine's Day and crap like that. So flowers are the sexual structures, and the reason flowers are pretty is because a lot of angiosperms are pollinated by animals, and they serve as a means to attract the animals. That's also why a lot of angiosperms also have a scent to them. They're trying to attract some kind of animal vector to move their pollen grains from the male structure to the female structure. Okay, these are different species, so that's not how it would happen. But to move their male pollen grains, their male gametes, to their female structures. And let's talk about the structure of a flower. You're definitely going to need to know this, okay? So we have a model that we looked at, and we also had an image in our lab manual. So either a picture or this model could show up again. So let's go over these structures. So starting from kind of the outside in. So the base of the structure, the base of the flower is called the receptacle. The first sort of whirl of tissue we encounter as we work our way from the receptacle are what we call the sepals. These can be leaf-like in appearance. Sometimes they're petal-like in appearance, but we're going to use a really obvious example where they're leaf-like in appearance. These protect the flower when they're in bud. Sepals collectively are sometimes referred to as the calyx. The next whirl in are known as the petals. I think we all know what petals are, but I have had people get that wrong on the lab exam, okay, guys? So also remember, I know sometimes it can be like stressful, but remember to use your common sense, right? Because you shouldn't get that wrong. Petals collectively are sometimes called a corolla. That's when you're talking about all the petals at once. You'd say, oh, the corolla is made of 5 petals. All right, the next whirl in are known as the stamens. These are the male structures. Okay, so here is a stamen. It's got 2 parts. The stalk is known as the filament, and the top piece is known as the anther, and that's where pollen grains will be produced. Interior to that is going to be the female structure, known as the pistil, sometimes also called the carpel. I'll accept either. 3 components: a stigma. This is usually wet or sticky. This is sort of the landing platform for pollen. The elongated style, this is what the pollen tube will grow down. And then the ovary, which we would find the ovule inside of. The ovary will ripen to become the fruit wall, and the ovule inside it will become the seed. So let's look at these structures up close. So imagine that we've taken a cross section right through this ovary wall. Here's what we would see. So this happens to be a lily ovary. And what we have here are basically, this is what we call a compound pistil made of 3 separate carpels. So it's a result of kind of a fusion of 3 fertile structures. Within each of these segments of the ovary, we find 2 ovules, so 6 altogether. So we can also call this the megasporangium, and we will be producing the egg in here. Let's imagine we take a cross section through one of these anthers here. What are we going to find? We're going to find, in this case, 4 chambers. Each of these chambers we would call a microsporangium, and within here we're going to find the male gametophytes, aka pollen grains that produce the sperm. Okay, so all of those structures are important. Okay, so we're now to our last lab, lab 12, which is all about structures of roots and stems. And one of the big things that is a goal of this lab is distinguishing between what we call a monocot and a eudicot. So these are 2 very large clades within the flowering plants. And I'm going to jump ahead just a second because there are several really good characters that help us distinguish what we call the monocots from the eudicots. And the reason they get their name, something called a cotyledon is an embryonic seed leaf. So when this embryo is in early development, it usually has these embryonic, large, sometimes fleshy leaves that we call cotyledons. And in the monocots, we just have one of them, and in the eudicots, we have 2. So a typical monocot would be something like corn. Think about a kernel of corn. You can't, if you were to try to dissect it, there really is no, right and left half. Now compare this to something like a lima bean or a kidney bean. That's a eudicot. You know how you can kind of like take a kidney bean and you can separate it into two sides? That's because it's got two cotyledons. It's a eudicot. So this pattern in embryonic development is how they get their name. But there are other features that are going to differ in monocots versus eudicot. And that's another important thing we need to keep track of in this lab. Okay. So stem structure. So again, everything is phylum Anthophyta here. We're talking flowering plants. So whether you're a monocot or eudicot, you're still a flowering plant. So there's some important differences in stem structure in monocots versus eudicots that you'll need to be able to recognize. So first of all, if we look at the stems, we see in a monocot, the vascular bundles are scattered throughout the stem. In a eudicot, we have our vascular bundles in a ring. The vascular bundle is a little hard to see here, but we typically have, oh, I have it up close. Much better. Okay, so let's look at our vascular bundles, monocot versus eudicot. Vascular bundles, so here's our phloem here, here's our xylem, here's our phloem here, here's our xylem, so you should be able to identify both the xylem and the phloem in these vascular bundles, as well as their general arrangement in monocots versus eudicots. Oh, I'm missing a slide. Where's my root structure slide? Okay, for some reason my root structure slide is not on here. I'll get that fixed, but for purposes of, you can also see it in the lab PowerPoint itself, which I'm going to put up there. So I meant to mention in the eudicot stem, we call this central area here the pith. This is these cells that are interior to the vascular bundles. And actually, I'm going to go find a picture of root structure. Okay, there we go. I don't know why that wasn't on there before. Okay. Whoops. Let's fix that. Okay, let's talk root structure. So, pretty different in a monocot versus eudicot. So, and by the way, eudicots used to just be called dicot. So if you see dicot, it's just they haven't updated the terminology. So in the eudicot, we have our xylem and phloem in this central area. We call this the stele, by the way, S-T-E-L-E, the stele, the central cylinder of vascular tissue. And basically what we have are these, the red are xylem, kind of in a cross shape, and we have patches of phloem within them. By contrast, what we have going on with the monocot root is we have kind of these circles of vascular tissue. So what we have going on here is we have sort of alternating xylem and phloem. So xylem is to the inside and phloem is to the outside, and they're kind of in this circle, and so we have some pith in here. By the way, tissue that we have to the interior of the vascular tissue is known as pith, and then when it's to the exterior, we call it cortex. So this has both a pith and a cortex to it. So you should be able to identify both the xylem and the phloem in a monocot and dicot root and a monocot and dicot stem. And let's see, I already talked about that. So let's talk about a woody eudicot. Oh, there's my root structure slide. I don't know why this is over here. So, okay, here's a much better picture. Oh, I like this one much better. Okay, so here's a close-up. So we have our phloem here, and we have our xylem that's kind of in this cross shape. That's the eudicot. And then in our monocot, we have our alternating patches of phloem with our xylem. And xylem is usually larger in diameter. That's one thing that will help you identify it. Okay, so I don't know why my slides are all out of order. I apologize for that. I'm going to fix that. You also need to know a woody eudicot stem. So some eudicots are what we would call herbaceous, meaning they don't make wood, and some eudicots do make wood, and we would call them woody eudicots. All monocots are herbaceous. Monocots do not put on woody tissue. So a couple of features you need to be able to identify. One is known as the vascular cambium, which would be running right here. The vascular cambium is responsible for putting on secondary xylem to its interior and secondary phloem to the exterior. So because xylem is much wider in diameter, the bulk of what a tree is, aka wood, what we'd call the wood, is actually the secondary xylem. What do I mean by secondary xylem? Well, the xylem that we make through primary growth would be our primary xylem while the plant is just growing up. And then when we're putting on secondary growth, aka growth and girth, we call that secondary xylem. And that's what makes up the components of wood. We also have what's called a cork cambium here. This is another kind of lateral meristem, giving us growth and girth, and it's putting on cork cells to the outside only. And this is what is going to make up part of bark. Also, secondary phloem is also usually considered a component of the bark. So usually this layer of cork cells that we call the periderm, this tough outer layer, plus the secondary phloem, we call that bark. You also should be able to identify an annual ring. That's pretty easy because we've been doing it for a long time. So I'm sure you know if you take a woody stem in cross section, you can count these annual rings. Well, the thing that makes up these annual rings and makes them easy to see is that secondary xylem, a year's growth of secondary xylem, can be broken up into what we call early wood and late wood. In the early wood, it's earlier in the year where usually water is more relatively available, and so our diameter of our xylem is much larger, and so when we look at it in cross section, it's lighter in appearance. In the late wood, there's less water, our xylem is smaller in diameter, and so it makes this usually this really obvious kind of ring, so we can actually count and say, oh look, 1, 2, 3. This stem is 3 years old. So we know how old a tree is by looking at its rings, which everybody knows that, but that's why. Already went over that. So leaf anatomy, you should also know your monocots versus your eudicots. So here's our monocot. Here's our eudicot. Okay, so a couple things to be aware of on the leaves. And again, you should also know these structures on the leaf models we're going to have in lab. So you're looking at a cross-section of a leaf. Now in a monocot leaf, we have what's called parallel venation. So if we were to cut a cross-section of something with parallel venation, you're going to see multiple vascular bundles. There's probably going to be one that's a little bit bigger, sort of the midrib, if you will. It's going to be a little bit bigger in diameter, but you're going to see several vascular bundles that are all similar in diameter. Also, what we're going to see in the monocot leaf, we're going to see a structure. We're going to see tissue known as mesophyll, and mesophyll are the cells that contain lots of chloroplasts and do the job of photosynthesis. In the monocot leaves, we just have one kind of mesophyll. Now let's bounce over to a eudicot leaf. In a eudicot leaf, we have two kinds of mesophyll. We have what we call palisade mesophyll. These are those barrel-shaped, tightly packed cells. And we have what is called spongy mesophyll that has a lot of air gaps in it for gas exchange. So you need to know those two types of mesophyll. Also, on our eudicot leaves, typically we only find stomata on the underside, whereas in monocot leaves, they can be both on the upper and lower side. So this is just kind of a nice comparison. So another couple things you should know about monocots versus eudicots is that in monocots, floral organs are usually in 3s or multiples of 3s, whereas in eudicots, they're usually in 4s or 5s or multiples thereof. Also, in addition to having differences in the internal structure of roots, there's difference in the external structure of roots in monocots versus eudicots. Eudicots typically have what we call a taproot, where you have one primary root and then thinner side branching roots coming off it, whereas monocots have what we call a fibrous root. Think of something like a grass, right? Ever try to pull up a grass? It's a real pain. You don't have that one single taproot. What you have are multiple roots that are of a similar diameter. Okay, so finally, fruits. So basically, you're going to need to be able to identify what kind of fruit you're looking at using a dichotomous key. So a little bit of terminology to remind you about. So the wall of a fruit is called the pericarp, and it's made of an endocarp, a mesocarp, and an exocarp. So if we think of something like a peach, the exocarp is that outer thin skin. The mesocarp is that juicy, thick part that you like to eat, that fleshy part. And then the endocarp is actually the wall of the pit. If you were to split a peach pit open, you would actually find the seed inside it. So this is a fruit type that we call a drupe. And what makes you a drupe is having a fleshy exocarp or a soft exocarp and mesocarp and a stony endocarp. We also sometimes call these stone fruits. Something like a plum is also a drupe. You have a nice description of these in your lab manual on your dichotomous key. So you need to know those parts of a fruit in order to use your dichotomous key. You also need to understand the distinction between fleshy and dry. If it's juicy, that means fleshy, right? Thick and juicy, something like a plum. Dry means that that ovary wall never got thick and juicy. Also, you need to understand the distinction between dehiscent and indehiscent. So our dry fruits, like nuts, like legumes, these are both dry fruits, right? We don't have juiciness in that fruit wall. If you are dehiscent, it means that you split open at maturity so the seeds can come out. If you are indehiscent, it means that you do not split at maturity. And you're usually a big pain to get open. All right, a little bit more fruit terminology and then we're done. You also need to know the difference between simple, aggregate, and multiple. So simple means that it's from one pistil on one flower. So we're talking about a flower that has one pistil and this ovary wall of the pistil got, fleshy or somehow ripened to become the fruit wall. If you are aggregate, it means that you're a single flower, but you have multiple separate pistils. And each of these pistils ripened into a separate little fruitlet. So a raspberry is a great example of this. A raspberry is actually an aggregate of drupes. So the raspberry, you know how you can kind of make a raspberry come apart into those separate little balls. Each of those little balls is from a separate pistil. So each has its own little seed. A multiple fruit is the result of many flowers on a branch that have all fused together. So a pineapple is a great example of this. The reason there's all that annoying leafy tissue in the pineapple is because we have multiple flowers and sometimes some leaf tissue gets in there that fuse together. And then finally, an accessory fruit. An accessory fruit is when you have tissues other than the ovary wall which are ripening and becoming fleshy. So a apple is a great example of this. So in an apple, the ovary is actually sunken down into the receptacle. And the ovary wall itself does not become fleshy. It is the receptacle that becomes fleshy and is the part of the apple you like to eat. The ovary wall is that really annoying leathery part that makes up the core that you don't want to eat. Another example of an accessory fruit would be a strawberry, right? The actual part of the strawberry you like eating, that is a swollen receptacle. And then the little things on the strawberry that you think of as seeds are actually fruits called the achenes, dry fruits called achenes. So a strawberry is an aggregate of achenes. Okay, oh my gosh, that's the end. Can you believe it? So I'll end off there. I'm also going to go over some labs so we can get a little bit more info. So I'll talk to you later.